Dr. Sage here, back discussing antimicrobial treatment. In this video, we're going to discuss the interactions between drug and microbe. By the end of this video, you should be able to explain the concept of selective toxicity, describe the 5 major targets of antimicrobial agents, and list major drugs associated with each, and identify which categories of drugs are most selectively toxic and explain why. Selective toxicity is a central concept in antibiotic treatment. Antimicrobial drugs should kill or inhibit microbial cells without simultaneously damaging host tissues. The best drugs in current use block the actions or synthesis of molecules in microorganisms, but not in vertebrate cells. Drugs with excellent selective toxicity include penicillins, which block the synthesis of the cell wall, which is only found in bacteria, not found in human cells. The drugs most toxic to human cells are drugs that act upon a structure common to both the ineffective agent and the host cell. As the characteristics of the infectious agent become more similar to that of the host, selective toxicity becomes more difficult to achieve, and undesirable side effects are more likely to occur. Metabolic targets of chemotherapeutic agents include inhibition of cell wall synthesis, inhibition of nucleic acid, DNA, and RNA structure and function, inhibition of protein synthesis, interference with cell membrane structure or function, and inhibition of folic acid synthesis. So the primary sites of action of antimicrobial drugs on bacteria cells include the cell wall, which is an excellent target because bacteria has cell walls, but your cells, animal cells, do not have a cell wall. These include things such as penicillins and vancomycin. We also have antimicrobial drugs that affect the plasma membranes, such as the polymycins. Antimicrobial drugs that affect the ribosomes, either the 30S small subunit or the 50S large subunit. Again, these can be highly selective because eukaryotic ribosomes are not the exact same structure as prokaryote or bacterial ribosomes. The ones that affect the 30S subunit are things such as tetracyclines. Things that affect the 50S subunit are things such as chlorophenicol. We can have antimicrobial drugs that affect metabolic pathways, for example, ones that affect folic acid synthesis, such as sulfoamines, or mycolytic acid synthesis, such as isoniazid. And finally, we can have antimicrobial drugs that affect DNA synthesis or RNA synthesis. For DNA synthesis, we can have ciprofloxacin as an example, or for RNA synthesis, we can have rifampin. Alright, this has been a brief overview of the interactions between drug and microbe. Until next time, this has been Dr. Sage.